Hi everybody, this is Alexandre White-Brown here again for another video for the SHINE Syndrome Foundation. I want to start off this video again by mentioning that these videos are only meant to provide some basic information regarding SHINE syndrome. These are not meant to replace any advice from your medical geneticist or any medical doctors that you might be seeing. So in my last video, I talked a little bit about the PSD95 protein and how that plays an important role in brain development and brain communication. Today, I wanted to answer a common question that a lot of families affected by SHINE syndrome have, and that's the different types of variants found at the gene level and how those affect the protein down here. So I'll start off today's video by going through once again what we call the central dogma and how we get from the gene to the protein and why that process is so important. Then we'll go through the different types of variants and show how a change at the gene can have such an important impact on the protein. So this can be viewed as a small snippet of a gene. So let's say the DLG4 gene, and there's just a specific code that makes up genes. This is 4 letters. We have T, A, G, and C. So T's are always matched up with A's, and G's are always matched up with C's. And this goes along the gene and provides a very important code. And these letters are strung together in a very specific way as they are what code the RNA. So we go from our gene to the RNA, and that code in the gene is what dictates the code for the RNA. So the letters for the RNA are very similar, except instead of having a T, we have a U. Now, for the RNA, these can be seen as being strung along in groups of 3, just like this. And these are what we would call codons. Now, codons are groups of 3 letters, so a code that encodes a certain amino acid. So there's several different amino acids, and depending on the codon in the RNA, that dictates what amino acid it's going to produce next in the protein. So this codon would create one of these amino acids. This amino acid strung along with other amino acids makes up the protein. And that dictates how the protein folds, its structure, and its ability to function properly. All right, so let's start off with discussing a common mutation called a missense mutation. Mutation can also be called a variant. Those words often interchange with each other. So if you hear variant or mutation, it's often meaning the same thing. And we'll start off by discussing a missense mutation. So when we have a missense mutation, what's happening is there's a substitution of a single nucleotide. So one of these is being changed, which means one base pair. So this grouping is being changed. So for example, we have C here, and we expect it to be a C. We are reading the gene in a certain way and would expect this part of the gene, this code, to be a C. But let's say, for example, it gets changed to an A. Subsequently, the G will get changed to a T. So now when the RNA is coming to read the gene, it's expecting to be reading a G, however, it's been substituted by an A. And there's many different forms of DNA damage that can cause these specific mutations. Oftentimes they're just by chance. Sometimes they can be inherited. In the first videos that I did for SHINE syndrome, it goes through a little bit about the inheritance and how these can be passed on. Anyways, we have an A here instead of a G. So this up here is going to have a consequence all the way down to the protein. So instead of this proper codon being created by the code up here, we may have a different letter here. So we're changing the codon because it's reading a different code up at the gene level. So instead of a U there, let's say, for example, it gets changed to a G, and now we have CGG instead of CUG. Now, since this codon has changed based on the change at the gene level, so the mutation causing a change at the RNA level, this codon is now going to code for the wrong amino acid. So let's say, for example, this codon is spelled in a way that's supposed to code for arginine, or here we have isoleucine. Since this has changed, this amino acid is going to be different. For example, let's say we might have glutamate. And the names of these aren't really super important. There are certain videos that you can go and watch in literature that gives you more information about the different types of amino acids and their different polarity and structure. But just for the sake of keeping this in layman's terms and simple, it won't matter too much about the specific amino acid. The important thing to keep in mind is that there's a substitution at the gene level. So if we go from a G to an A, this means that when the RNA is being coded, we have a change in the codon, and this is coding for a different amino acid than what we would expect. Now, for missense mutations, the protein is not truncated. So you've probably heard the term truncated protein, and that means that the protein ends up smaller. I'll be discussing a little bit more later in the video about frameshift mutations and nonsense mutations, which can lead to a truncated protein. But for missense mutations, we get the same size of protein. So it's not stopping the protein from being made. All it's doing is changing an amino acid. So on your genetic testing results report, you might see a C dot number and then a letter change. So let's say C to G. And this shows where in the gene and what letter is being changed. And then preceding that, you might see a P dot, and this is explaining what amino acid is being changed. So you may see arginine, and then you'll see a number, and then you'll see another, let's say, glutamate. So it's being changed from arginine to glutamate. So for example, here, this codon is now coding for glutamate instead of arginine. And you may think, is it very important that one amino acid is being changed? How can that have such an important impact on the protein? And sometimes for missense mutations, it doesn't have a big impact on the protein. And the way that we look at this impact is we look at how it affects the structure of the protein and how it affects the function of the protein. So protein and all these different amino acids strung together, they all have certain polarities, and they are all strung along in a certain way that allows the protein to fold properly into a way that allows it to function properly once it gets to the synapse, allowing other proteins and other mechanisms to function properly, as discussed in previous videos. However, if we have a new amino acid that has a very different polarity, or that may change the way that the protein folds, it can have a serious impact on the structure of the protein. Now, if we have a protein that doesn't fold properly based on the structure of the protein, this can signal to the cell to degrade that protein. It's not useful, we should get rid of it. So that may trigger a response to have the protein degraded, or if the protein does not get degraded, it may get to the synapse but not function properly. And this can have an impact on the other proteins and other mechanisms at the synapse, allowing it to not function the way that we would expect. So when we think of the impact of a change up here to the RNA and the change in the codon, this has an impact on the amino acid that's strung along to make the protein, and this can have an impact on the structure of the protein, and or it can have an impact on the function of the protein. Important to keep in mind too, once again, that for a missense mutation, the protein is not truncated, it's staying the same length that we would expect. It's impacting the way that that protein folds and the structure of that protein and how that protein functions once it gets to the synapse. The next type of variant that we'll discuss is what we call a nonsense variant or mutation. This can also be called a stop-gain mutation, and I'll explain a little bit why as we move along. So a nonsense mutation is very similar to a missense mutation in the sense that a single nucleotide is being changed. So we have a single base pair that's being changed. However, the consequence of that single nucleotide change ends up with the RNA stopping early, and therefore we end up with a truncated protein. So once again, for example, let's say our A here gets changed to a C. Subsequently, we have a G up here. So this is our change here. We have our single nucleotide change there, and this is going to be a nonsense mutation. And the reason is because this change is going to lead to what we call a stop codon, and this is going to be a premature stop codon. So usually what happens is we have our code that goes all the way along, all the way down the gene, and then we have our RNA, which is coded that goes all the way to the end, that codes for the perfect amount and the perfect size protein that we would expect, and that is functional and able to be used properly in the synapse and in the cell. At the end of the RNA, at the end of the protein, there's what we call a stop codon, and this tells the RNA and the gene and the protein to stop because we've reached the length that we need, and it doesn't need to produce any more amino acids, and the polypeptide chain, the protein is finished. It's ready to be cut off and done. What happens with a nonsense mutation is that we have a change here, similar to a missense mutation. However, this is causing a premature stop codon. So there's a specific 3-letter code that tells the RNA to stop producing the protein. And this is UAG. So if we have a change in the letters up in the gene, a change in this code that accidentally codes for the UAG codon early, this is basically creating a big stop sign here. So this is stopping early. Instead of continuing to read on and continuing to code for the protein, it thinks it's supposed to stop right here. What happens is this then creates a stop here, and it stops producing more amino acids and building the PSD95 protein. So what we end up with is a truncated or a smaller protein, just like this. It's stopping here, and there's no more protein, no more amino acids being added on to the protein to make it the proper length. So almost always a nonsense mutation is going to have an effect on the structure of the protein. It's not going to be as long as we would like. And once again, almost always is going to have an impact on the function of that protein. So with a truncated protein, it's not going to fold properly. It's not going to be structured the way that we would want it to be, the way that the cell thinks it should be. And oftentimes, once again, this leads to degradation of the protein. So it gets degraded once the cell realizes and has a signal that this is not right, this is not what we expect. And then if it is able to make it to the synapse, it is not functioning properly. So oftentimes with a nonsense mutation, we have a protein that is structurally and functionally not proper, and therefore we have a non-functional protein and oftentimes what we call a damaging mutation. So the next type of mutation that we'll discuss, and this one is a little bit tricky to understand, so we'll try and go through it slowly, is what we call a frameshift mutation. Now, a frameshift mutation is a little bit different from missense and nonsense mutations, as what's happening is there is an insertion or a deletion of the nucleotides that makes it so the RNA cannot read the gene properly. And what we end up with is several amino acids being coded wrong. So we end up with the wrong amino acids being strung together. And sometimes this can also lead to a premature stop codon similar to a nonsense mutation. So when a gene is being read, it is being read in frames. And these frames are in sets of 3. So I drew this here to really show that we have a group of 3 here, we have a group of 3 here. We have 3, 3, and this would be the reading frame. So this would then code for 3 here, and this is once again the codon. So it's reading along, it's coding for the RNA, and it has these groups of 3, and they're expected. It's how we want to read it. It's making the right codons, and that's producing the right amino acids that are being strung along for the protein. But then what happens if we have an insertion? So let's say we insert a nucleotide here, and let's say it's a G, and then this would make it a C up here. Instead of reading the proper reading frame from T, C, and A, it's now reading these 3 together. So T, C, and G. This then has a subsequent effect on the rest of the reading frames. So instead of G, T, and A being read together here in its reading frame, we instead have A, G, and T, and so on and so on. So this is creating a domino effect of reading frames that aren't being read properly. And as you can imagine, that means that this codon is being read wrong, because instead of TCA making the proper codon, it's now TCG making a different codon, and so on and so on, all the way down the gene. So what this means is that the proper code for the amino acid is subsequently being produced wrong. So it's creating a protein, it's creating a polypeptide chain of these amino acids, but they're not the amino acids that we would expect. Because we have the reading frame as different here, it's making a different codon, which is making a different amino acid. And this is a subsequent change all the way down the protein. What this leads to is a protein that's not structured properly. So it's not the amino acids that we would expect. This doesn't allow the protein to fold and structure itself properly. And most times with a frameshift variant or a frameshift mutation, we end up with a non-functional protein. So a protein that cannot be used properly to do what it's supposed to do. And going back to the previous video about PSD95, the nonsense in the frameshift mutations that lead to these non-functional PSD95 proteins can have a very big impact on brain communication. So I gave an example here of an insertion, but another example is we could have a deletion. So we could have 1 or even 2 of these deleted. So it's a deletion of the nucleotides. And what this is doing is it's still changing the way that these are read. They're always going to be read in 3. So it's subsequently changing what 3 nucleotides are being read. And then this in turn will change the codon. So these 3 will all change. Let's say A, C, and G instead of what it was. And then this will subsequently change the amino acid that was there. And once again, this will have a subsequent effect all the way down because it's reading by 3 and it's shifting. It's shifting the frames that it's reading, and therefore the subsequent amino acids creating the protein are all changing from what we would expect them to be. Now, it's possible also that a frameshift variant could lead to a premature stop codon similar to a nonsense or stop gain mutation. This is because the domino effect could lead to a UAG, and this would stop the RNA from coding the protein, and we would end up with a truncated protein. So oftentimes for a frameshift mutation, we end up with a non-functional protein, a truncated protein quite often, and in the end, a protein, PSD95 protein that's not able to do what we would want it or need it to do. All right, so the last type of variant that I would like to discuss is what we call a splice variant. Now, this type of variant can be a little bit confusing, so I'd like to start off by discussing what splicing is. So up at the top here, we have a very zoomed out look at the DLG4 gene. So the boxes with numbers are what we consider exons. And if you think back to the first video, I mentioned that DLG4 is a 22 exon gene. So there's 22 exons that are the coding regions for the RNA and then eventually for the protein. But there are parts of the gene that are considered non-coding, and those are these little strings in between the exons, which are called introns. So the way that you can think of this is these are like beads. So the exons are like beads on a string, and the string in between the beads can be considered the introns. Now, as we move down from the coding of the gene down to the RNA, there is an important mechanism called splicing that gets rid of the middle part, the introns, so that all we have left as we move from our RNA product that's ready to produce a protein is the coding regions. So as you can see here, we have the introns at the gene level. And then as we get through our RNA product, what we end up with is the exons all together as a long coding region. So this means that we're getting rid of this intron and this intron and this intron, and we're basically bridging the gap so that we have our RNA product. And that RNA product, once strung together, can start to form and encode the proper amino acids for the protein. So a splicing variant is a variant that may occur very close to the exon and intron boundary. So it may be in here or in here in what we call the donor or acceptor sites. And this can have a large impact on what we end up with here. So one example is if there is a mutation in the area that can affect splicing, one thing that it can do is mean that we end up with pieces of the intron in this RNA product. So for example, we would end up with, instead of having this perfectly connected, we may have exon 1 here, but we also have a piece of the intron still in that RNA product that's going to be coding the amino acids for the PSD95 protein. And that can have an impact on the overall protein product as it's not being able to code exactly how we would expect, similar to other mutations. Now, another type of problem that we can incur from splicing that goes wrong, put the one back there, is that this can sometimes lead to exon skipping. So this means that as it's reading along and trying to create this proper RNA product, if there's a variant in a place, in one of the boundaries, the acceptor or donor splice sites, it can lead to perhaps a skipping of an entire exon. So what we would end up with may be the loss of exon 2 in the RNA product. Now you can imagine that without that exon, without the coding within that region, we're going to end up with changed amino acids. So let's say histine to a glycine. And then we're going to end up with a protein that we were not expecting. And this can lead to a insufficient PSD95 protein that's not structurally sound, may not be functioning the way that we want it to be. Now, the last consequence that we can have from splice site variants is that sometimes we have the proper, we have our 1 back, we have our 2 back, but then sometimes we can also have smaller parts. So we have our full exon 2 that's been spliced properly, but then we only have half of our exon 1. And this can be because perhaps there's a splicing variant that only allows part of this exon to be read, and then we only end up with a partial exon 1 that's spliced properly in the final RNA product. And this, once again, will have a consequence at the protein level, leading to a dysfunctional protein or a change in the structure of the protein, which will end up affecting how the PSE95 protein can interact with other proteins at the synapse level. So those are the different types of variants or mutations that I'm going to go through today. There are other types of mutations or variants, such as intronic variants, that are a little bit more rare and not as common as the variants that I described today. So if anybody has any questions or comments with regards to variants that I didn't touch on today, please don't hesitate to leave a question or a comment, and I'll try to respond to it as quickly as possible. I wanted to end today's video by reiterating that Shine syndrome is inherited in an autosomal dominant fashion. So if you remember from the first video, we received one copy of the DLG4 gene from our mother and one from our father. So we have 2 copies. To cause DLG4 Shine syndrome, only one copy needs to have a damaging mutation. So in the end, we have a fully functioning copy of DLG4 that's able to properly produce the PSD95 protein to the size and to the capabilities that we need it for. The other copy, however, may have a damaging mutation, whether it be a missense, a nonsense, a frameshift, a splice variant, or a different kind that is harming the way that this gene produces the protein, and that can be what causes Shine syndrome and leads to what we call haploinsufficiency, a term that you might have heard before. So I'm going to end the video there today. Once again, I just want everybody to know that if you have any questions or comments, please don't hesitate to leave them. I'll try to respond to them as quickly as possible, and I'll see you soon with another video.